Hello. In our studies, we're continuing today from level that we have attained the cellular level of organization, to the next level of organizing the human body, which is the level of tissue. So before we start, maybe more of a rhetorical question would be asked if you can just quickly answer how many different types of tissues you believe is composing the human body. I intentionally made here a little bit of a stop, just trying to give you at least a couple of seconds of time to think about and to come up perhaps with a very specific number. I wouldn't be surprised that numbers that you currently contemplate being correct answer are going 7, 8, 10, maybe 15, because practically lots of people cannot just wrap their mind around the idea that the human body is only composed of 4 basic types of tissues, just 4. And if we're to list what these 4 categories are, basically we have one large group, which consists of what we call the epithelial tissues. Epithelial tissues are practically those ones that are seen covering the body's surface, and of course the skin comes as the first and best example of it, but also epithelia are lining up the internal surface of hollow organs, such as lining of the blood vessel, such as lining of the respiratory tract, such as lining of the digestive tract. Basically, the inside of these hollow structures are also covered with epithelium. Certainly, there is a huge difference between the epithelium lining digestive or respiratory tract versus epithelium covering the surface of the body, the skin, but they fall under the same category. Also, into this part of our studies, we would have to include the glands, which have the exocrine secretion. What is meant by exocrine secretion? Glands are of epithelial origin, and if they secrete and release their products either onto the body's surface or into cavities of organs, basically they would be deemed to have quality of exocrine secretion. In contrast to exocrine glands, there's another category, which is called the glands with endocrine secretion. Endo, which obviously means inside, applies to glands that release their product directly into bloodstream, and then the bloodstream will carry their products to designated parts of the body, the so-called target tissues, where the effect of substance produced by endocrine gland will ultimately be reaching its goal and will reach the target tissue, and action of that substance would be observed. Second group is also quite large. So after epithelial tissues, we're coming to a group of connective tissues. This is also where we're going to have unbelievable varieties, where we're going to go from connective tissue that, technically speaking, is fluid. It's a blood. It is categorized also as the connective tissue, but then we can go to opposite end of a scale and find something that's a little bit more firm, a little bit more tangible, a little bit more supportive, and we will find and fit in that bone as a tissue and the cartilage as the tissue is just a subclass of connective tissues that we're finding within the body. Adipose tissue or fat is yet another example of connective tissue, which is neither liquid nor solid, but rather something that fits physically in between. And that's why the epithelial and the connective tissues will be presented in their individual video sections of this series. Today, we'll be talking about epithelial tissue. The next video will address connective tissues. And then the next 2 videos will address remaining 2 types of tissues, which we expect to have within a human body. One of them the third one being the muscle tissue, and the fourth and last one will be the nervous tissue. So might be a little bit of surprise that we're that simple, just being made of 4 different types of tissues, but within each type, we're going to find some kind of variances. And at the end, of course, we will not be seen as being extremely simple in building up the human body. So what do we say in general about tissues? Well, tissues are groups of cells of the same kind that are linked together in order to fulfill the common function, which is assigned and allocated to them. One of the critical things that we will have to address, and it is already like in front of you on the screen, is that between cells that are forming tissue, we must establish some kind of physical connections that will prevent these cells being pulled apart, being separated. And of course, should that ever happen, there will be a disintegration of tissue and most likely significant loss of function, which cannot be absorbed and tolerated without some kind of massive impact to the rest of the body. So in the center of the screen, you have example which illustrates one single cell with apical projections, likely being called microvilli. And throughout that cell and its cellular membrane, you'd see those little small squares identifying A, B, C, D, and E different types of bonding, which could be formed between 2 adjacent or adjoining cells. Of course, we will not be able to see all 5 different types of bonding on each and every cell within the human body, so this is just for illustration purposes. That would help us understand a little bit better what is going on and how do we appreciate some of the differences between bonding 2 cells together, not just allowing their walls to stick together and putting some kind of adhesive substance between them, but rather different forms of adhesion actually are there to ensure a different quality that certain tissues produce. So we're going to go with tight junction being the first type of bonding that should exist between adjacent plasma membranes of 2 different cells. They look like little rivets, and they're really transmembrane proteins that exist from one cell across the intercellular space and then end up by crossing and sticking into a membrane of a cell which needs to be bonded. Just the word tight junction probably gives you more than enough information because there is no secret meaning in the word tight or tightness. Basically, the connection that exists between 2 cells is so strong that practically it prevents any kind of substances being freely passed between 2 cells. So that is the true meaning of this tightness, or any other content leaking, let's say, from inside of the body between epithelial cells to the body's surface, leaking freely. That is not going to happen. So this one being perhaps the most straightforward, we move on to the next one example, which is called the adherence junction. It is illustrated here under diagram letter B pointing out, so you can see about the location between 2 cells, but now we have to accept and to understand what is going on. On cells that require bonding of this type, adherence type of bond, as you can see, there is some kind of additional thickening within the inner aspect of cellular membrane. Basically, we see a location of multiple microfilaments, which are attached to this thickening that is called the plaque. Microfilaments, as we remember, are part of the internal composition of the cell, something that ensures that internally cell appears to be arranged and organized rather than disarranged and disorganized. So on the plaque from the outside and going through the cellular membrane, so like transmembrane protein, we have proteins that are called cadherines. And both cells requiring bonding will have their own cadherins that will be projected into tiny cleft between two cells, so-called the intercellular space. They would quite tightly overlap with each other. And if you're looking for a down-to-earth example of what might be comparable to adherence junction, is a Ziploc bag. Very similar. We know that there is some kind of thickening form of plastic between front and back of the bag, and when we squeeze these two thickened parts, we hear little clicks as the interlocking occurs, and the bag becomes pretty much quite firmly closed. Why do we need adhesion belts? Well, we need them also to prevent any kind of separation between two cells. A good example would be cells of our skin, which simply, if they separate, they would widen the gap and there would be an entry point into our organism. Similar bonding could be seen also between heart muscle cells, because under different pressure, blood coming in, filling in the chambers, and the heart contracting to eject the blood into arteries of the body, there will be lots of physical stress. And of course, two cardiac cells, two myocardial cells would prefer a solid and safe type of bonding, which will prevent their separation. The third example is what we call the desmosome form of binding. As you can see, there is a bit of similarity between what adherence and the desmosome binding would look like. We're going to have, again, on the inside of the cell, number of different intermediate proteins, mostly keratin, attached to relative thickening of the cellular membrane on the inside, again forming the plaque. From the plaque, transmembrane proteins, cadherins, again very same as what we have seen in the adherence junction, project into intercellular space, overlap, and create form of bonding, which doesn't have a shape of a strip going practically longitudinally, but rather it looks like some kind of bonding which could be sporadically observed between two cells that require bonding through desmosome. So we are to see another example, which is called the hemidesmosome. In this case, hemi practically means half, so really like half of the desmosome. And if we take a look, we're going to just find out that instead of cadherin, we're having protein which is of a different kind. It's called integrin. And hemidesmosomes are usually seen and observed at the base part of the cell. Practically semi or hemidesmosomes are going to bind a cell and make it inseparable from the underlying basement membrane. Otherwise, if this cell is example of epithelium, basically we don't want to see the situation where the epithelium separates from the underlying tissue, because the problems will be multiple, as we're going to see it already from the same next slide. And ultimately, one of the very uniquely found junctions between these cells is what is called the gap junction. Gap junction really looks here as some kind of indented part of the cellular membrane and some form of tubules that are running from cytoplasm of one cell across the intercellular space and into the cytoplasm of a cell which is bonded. The gap exists between cells, but these tiny tubes that are called connexons, made of protein called the connexin, are ensuring that any kind of change affecting cytoplasm of a cell on the left side of this diagram will go through these tubes and will start gradually affecting cell, which is seen on the right side of this gap junction. It is particularly important to remember that this form of bonding will exist between myocardial cells, but this time not for physical holding of two cells together, rather because we want electrical current, the charge which affected one cell at the beginning, easily spread out through the connexons, and then in a form of linear connection starts affecting other myocardial cells and stimulate them to do contraction. So back to just the idea that was shared not even a minute ago, hemidesmosomes and their role to ensure that cell at its basilar part practically will become firmly attached and inseparable from a basement membrane. And here is example of why is it so important. We have a group of 4 different cells representing epithelium. Through their side surfaces or lateral surfaces, they obviously attach to each other. It doesn't matter what kind of bonding we're going to have between cells. Neither it will affect us to understand what exactly is happening at the apical free surface, the one which is oriented externally to the surface of the body or into internal cavities of the hollow organ. What we immediately see as it's not there, it's direct supply. These cells need blood supply, and it is the quality of epithelial tissues that they do not have their own direct blood supply. Obviously, they have to rely on the underlying connective tissue through which a variety of different nutrients will be simply diffusing in the direction of epithelial cells. But if they're separated, the process of diffusion would stop. And if damage is done to epithelium, it would be irreparable and, of course, with ultimate negative effect to the rest of the body. Although epithelia do not have their direct blood supply, they do have direct innervation. And as we can see here, some kind of free nerve endings are sticking into spaces between multiple epithelial cells, which is a really great idea to share because our epithelium, skin in particular, is quite innervated, and we are able to use any kind of physical contact between different parts of our body and our environment in order to accumulate more information about us and to be able to plan our next step. Another good quality of epithelia is that they have a good capacity to repair the damage. As all of us would know from their own experiences, we're going to just recall last time when we had a little cut on our finger, which caused perhaps a bit of bleeding and need to put a Band-Aid. Clearly, the blood was coming from an injured tissues, but in a couple of days' time, when a scab is formed and the scab falls off, basically the gap which has been created by this kind of defect caused by either a knife or scissors or some kind of other sharp tool will be nicely filled in with normal tissue, and we are not necessarily to see formation of any scars, but rather the defect would be fully and properly closed. So major things to kind of take as a message here is epithelia are innervated, but they are avascular, meaning they don't have vascular support of their own. And good quality in repair is essentially what we also need to remember. Obviously, if we just have 4 types of tissues to compose a human body, epithelia, connective tissues, muscular and nervous tissue, one can ask a question, then what kind of differences are we to find within the different subclasses of epithelial cells? The answer is relatively simple. And if you follow this kind of schematic way of thinking, you would be able to sort out the rest of this presentation pretty much quickly and easily. First, we're asking ourselves, okay, what kind of arrangement of cells we have within tissues? And we have 3 possibilities, either to have a single layer of cells, as illustrated on this diagram, or we're going to have multiple layers. And in this case, we would say this is simple epithelium, single layer. And if multiple layers of cells are seen one on the top of another, then we're talking about stratified epithelium. Strata means layers, stratified epithelium. Sometimes we could have a little bit of a visual dilemma. What do we see with our eyes? And that's why we will have a chance to identify also pseudo-stratified epithelium, because the arrangement of nuclei will be seen at different levels within multiple cells, although it is a single layer. That kind of presentation of cellular nuclei would give us the false impression that we're talking about some kind of irregular multi-layer. Hence, the best name is pseudo-stratified epithelium. So that is the first guide that we can use. Single layer, multiple layers, or single layer, but looking a little bit disorganized, and we call it pseudo-stratified. Then another interesting lead that we can follow is what is the shape of the cells that we see forming the epithelium. Sometimes these cells are very flattened, just like floor tiles in our bathrooms or kitchens. Obviously, they have width. They measure what, 5 by 5, 10 by 10, 15 by 15, or any other dimensions, but the thickness of the tile is something that is much significantly less than the length and the width. So these cells, if they're flattened, we call them squamous epithelial cells. SQUAMOUS, squamous epithelial cells. Then these cells could become a little bit thicker. So each cell would look like a little cube. Of course, we're not talking about the length, the width, and the height being always the same, but they're coming close to being comparable to each other. And that's why we're then calling cuboidal cells to make this type of epithelium. And finally, the third, the tallest type of cells, where height obviously is greater than the length or the width, we call them columnar cells, and we have therefore three categories, either squamous, cuboid, or columnar. There would be a fourth category. Just fourth one will be seen at one particular area within a body, urinary bladder. We call it transitional epithelium. Epithelium that lines the bladder will change its shape on the basis of how full or how empty the urinary bladder is. When the bladder is full, its walls will be stretched, and naturally these transitional cells would look thinner, closer to squamous. Upon emptying of the urinary bladder, cells will recoil, and they're going to have shape which is much closer to cuboid rather than to squamous cells. And final, third category that we have to use in order to properly classify all the epithelia is, do we have any kind of projections that are eventually seen on the apical surface? Sometimes we could see projection of tiny villi, and we have to immediately think about what is it for. Basically, without any projections, this apical surface would be very directly and easily measured. So we would know that its surface area actually is what its physical length and width are. But with multiple finger-like projections, there will be a very huge increase of surface on the apex of the cell, which really favors processes of absorption. Larger the area that the cell exhibits to make the contact with the substance that we are looking forward to absorb, of course, is going to be made much simpler and much easier if the cell is ciliated. So I believe that with these kind of very technical ideas, squamous, cuboid, columnar, and then single layer, multiple layer, or falsely looking as multiple layer pseudostratified, we have two basic criteria that we can use and to classify rest of the epithelial tissues. So we, from this point on, will go perhaps a little bit faster, as you would have no need to really know this with a great amount of detail. However, I will try to give you a decent guidance through these slides and help you with your studies. Obviously, these are all glands. and they have ducts that will take the product of the gland onto the surface of the body. Based on their shape, glands that are exocrine also would be going through different classification. They could be looking like simple tubular, simple branch tubular, simple coiled tubular, simple acinar, simple branched acinar, compound tubular, compound acinar, and compound tubulo-acinar. So, although you and I have gone through all these different 8 types of exocrine cells, essentially what really makes the common denominator, regardless of how simple or how complex the gland looks like, and regardless of how many different parts the gland would be made of, what is common to all of them, that they produce the secretory duct, which opens up either on the surface of the body or it opens up at the lumen of the hollow organs. Since some epithelial cells, glandular in particular, are designed to secrete, and their products need to be taken out of the gland, we're to briefly address what we have here on this slide. There are 3 different types of secretions that epithelial cells could utilize in order to release the product they synthesized into the cellular or glandular space, into duct of the gland, and then into different parts of the body or into different organs. We know that this process is something that we're already familiar with, that through the process of protein synthesis, we would subject them to certain changes and packaging within endoplasmic reticulum or Golgi complex, forming the secretory vesicles that travel towards the apical parts of these cells, where the secretory vesicle blends with the cellular membrane, opens up, and simply releases the content out of the cell. That is the merocrine secretion. Apocrine secretion is slightly different, but in general, it's quite comparable to merocrine secretion. The glands utilizing apocrine secretion allowed their product to accumulate within the apical part of the cell. So the difference is that these transportation vesicles do not merge with the membrane opening up individually. Instead, the cell which uses apocrine secretion pinches its apical part off, and together with part of cellular membrane and cytoplasm, basically we're going to have those transportation vesicles that would contain a substance that needs to be released. Finally, the holocrine secretion is perhaps most sacrificing, because during its productive time, the cell simply accumulates its own product, which accumulates at the expense of cellular other structures, organelles, cytosol, including the nucleus. At one point, this cell practically accumulates sufficient amount of substance, which is synthesized, and the cell practically opens up, releasing its content. But as it happens, this cell also reaches the end of its life. However, cells that are located deeper within a gland in the meantime will be going through cellular division, and newly formed daughter cells will be replacing those ones that have been lost through the process of secretion. This slide will be using point for us to try to identify a variety of different types of connective tissues. So we'll come back to it in due time, but in the meantime, we want to continue exploring, and as I said, maybe a little bit faster, different types of epithelia, just to find out where are they, what are differences, and perhaps a few extra additional things that we might need to know about it. So we know criteria, how many layers, 1 or multiple, and what type of cell. Is it squamous, cuboid, or columnar? So what we see here is a simple squamous epithelium. Obviously, cells like tiles being very thin, neatly placed next to each other. No major gaps between these cells, directly attaching them to the basement membrane. And in a living human body, basically we have this type of epithelium lining inside of the abdominal cavity. There is a membrane which is called the peritoneum that lines the walls of the abdominal space, but also the very same membrane continues and covers surface of a number of different intra-abdominal organs. We call it peritoneum, and obviously it doesn't need to be of a great absorptive or a great secretive. It is more for physical protection, and this simple squamous epithelium in the form of peritoneum will do quite well. Here is another example of single layer, but this time being made of cuboidal cells. I love on this slideshow that Medical Illustrator created very easy to grasp handmade diagrams, just showing the differences between different types. Again, cells must be attached to the basement membrane, and under the basement membrane, there has to be some connective tissue with blood vessels. As we know, epithelial cells do not have blood supply on their own. Where do we find it? Here it is, obviously organ, part of the digestive system, the pancreas. And on a photomicrograph, you can see here these cells practically all perfectly lined and leveled, all being of a very comparable size and a position of their nuclei, also with some kind of like military precision being leveled, all at a nice same level within a cell. So simple in a cuboidal epithelium that is lining the ducts of pancreas. So this is the lumen of the duct, where pancreatic secretion will be further distributed into digestive tube. This is just like linings of the wall. So the next one must be single layer, but columnar cells, much taller than cuboidal. And here we are. Same idea, single layer of cells. Their nuclei are not always ideally leveled at the same spot. And as you can see between these tall columnar cells, we're going to have some additional cells. In this case, it's a goblet cell producing a mucus, which also needs to be excreted into the lumen of a hollow space, such as small or large intestine. These cells exhibit lots of cilia on their surface, which we know increases the absorptive area of the digestive tube. And by doing this, of course, we have greater accuracy of absorbing all the important ingredients we ingested through oral cavity, whether they're liquids or solids. Essentially, we need to absorb them and allow these different nutritional substances to reach into our bloodstream. Photograph is of sectional view, non-ciliated, simple columnar epithelium, which is lining the jejunum, that is one part of the small intestine. Other two parts are called the duodenum and ileum. And as you can see here, nuclei, here are some mucus goblet cells. Naturally, we have to have mucus which physically protects the inside of our small and large intestine. And this kind of blurry apical end is what 675 times magnification captured, but we know these are microvilli, which are increasing absorptive surface multiple times. So that would be three types. And now we're to see another example of ciliated simple columnar epithelium. This diagram looks very much like the previous slide that we used, except this time location is different, and it is the uterine tube or oviduct. Sectional view of ciliated simple columnar epithelium of the uterine tube. Obviously, cilia in this case are not for absorption. When the ovary releases immature ovum into oviduct, movement of cilia will actually facilitate movement of ovum deep into oviduct, where eventually, if the pregnancy is to happen, meeting point between male and the female reproductive cell will take place, and then conceptus, practically fertilized ovum, will be further pushed by same movement of cilia to pass the rest of the oviduct and to get into the uterine cavity, where it's going to implant and will stay there for remaining 9 months until the labor is triggered and a newborn comes. This time, we're talking about multiple layers, stratified squamous epithelium. And interestingly enough, the illustrator did not opt to say, let's go with the skin, but rather they used walls of vagina, part of the female reproductive tract, and within the purple colored part of this photo micrograph, you can literally observe tens of different layers of cells. There is something that's easy to remember. Those younger, newly formed cells appear to have the deepest position. But as the new cells keep continuously coming, they gradually push cells that were made yesterday by cells that are made today. And as this process continues, we're moving closer and closer to the surface, and cells, of course, become detached further away of underlying supporting connective tissue. They lose some of their physical qualities, which is like they started almost like cuboidal cells, but further away they go, they become more flattened, and then they simply exfoliate, they shed. So we have a constant need to keep replacing it. Just the very same thing affects our skin. We have cells that are coming from the surface of our body that are lost, and they account for most of the household dust. They are gradually replaced by newly formed cells, and the integrity of the skin practically just continues throughout our life. Here we have layers of cuboidal epithelium. Not too many of them, just 2. So this is the stratified cuboidal layer. Again, epithelial cells have to be attached to the underlying connective tissue through the basement membrane. And we're to observe appearance of this multi-layered cuboidal epithelium in the esophageal gland. So it's not the wall of the esophagus, because it would not benefit from just 2 arrangement of cuboid cells. Esophagus will have to have a different type of epithelium because we swallow, and not necessarily always we're swallowing very soft and pliable content. Sometimes we don't have enough time to chew, and we swallow something that with difficulties keeps passing through the esophagus, eventually sometimes even scratching its surface. So this is really just a duct of the esophageal gland, which is made of 2 layers of cuboidal cells. Stratified columnar epithelium. So we're talking about columnar cells, but multi-layers. Again, because this is epithelium, presence of connective tissue in the basement membrane, which supports the epithelium, is what we need. And now you can see sectional view of stratified columnar epithelium, also a duct of the esophageal gland. Lumen of the duct is here, and you can see, and without any difficulties, at least counting 1, 2, 3, or maybe over here 1, 2, 3, 4 different layers of cells that are clearly not just cuboidal, but rather more elongated columnar type of cells. And here is that interesting transitional type of epithelium, the one which lines the urinary bladder. And I already explained that type of epithelium, to be called transitional, is the epithelium which really keeps going through very physical stress each and every day, multiple times, by accumulating urine within the bladder, stretching its wall, therefore stretching the epithelium. But upon bladders emptying, the epithelium will recoil and regain more or closer to cuboidal shape when empty, and when full and completely distended, basically the cells will remind you of squamous stratified epithelium. What was said, when we have a single layer, but with really disorganized positioning of the nuclei, this type of tissue could easily mislead us to believe that it is stratified layer of columnar ciliated epithelium, where the essence is that it is a single layer. These cells are very tall, and even on this photomicrograph, 300 times magnification, basically we would be misled to say that there are multiple layers. Dark spots are nuclei that are captured at different levels, lots of cilia on the apical surface. Again, it's the respiratory tract, which could be contaminated by a variety of different micro particles that will be trapped by the ciliated epithelium, and a movement of cilia will keep moving it further up until it finally accumulates in a sufficient amount that it triggers cough reflex, or sometimes, as when people have to speak for extended period of time, they would simply say, clear their throat and continue talking about what they are presenting. So this is pseudostratified ciliated columnar epithelium. Endocrine gland is a gland, and we mentioned that we're talking about glands with internal secretion. Their secretion parts will be sometimes thrown into little ovals or globular structures that we simply call the follicles, and the type of epithelium is what we can compare and say this looks like single cubital epithelium. And these cells, instead of secreting into secretory duct, would rather allow their product to be collected within the cavity of the follicle itself, and when released, it will be released into bloodstream. So for all the glands with endocrine secretion, we simply say they are to release their product into bloodstream rather than to see it being released onto the surface of the body. Exocrine gland, like sweat gland, we can see here definitely two layers of cells, lumen, which accumulates the product, and then you can see the long secretory duct that'll take sweat all the way to the surface of the body, where it's going to be released and made physically visible. At this point, I believe that we have made fairly decent introduction to different form of epithelia that exist within the body, and we can now move on in our next video to present a variety of connective tissues. I really prefer to separate them, otherwise the recording itself would become quite long, and I don't think that there would be appeal for any one of you guys to sit in front of computer and watch the single presentation for over 1 hour. So see you soon in connective tissue sections. Thank you for watching this video.